Hi there, and welcome back to our section on macromolecules. We are officially beginning our macromolecule exploration, if you will. So we have 4 macromolecules to study. The first is carbohydrates, the second will be lipids, then we'll follow it with proteins, and then nucleic acids, which are your DNA and your RNA. So we left off studying Roman numeral 3. We were in macromolecule reactions, how you take monomers to build polymers, and how polymers can be broken down into monomers. So let's talk about those different types of functional polymers called macromolecules. The first one, carbohydrates. So Roman numeral 4 is carbohydrates. And what a carbohydrate is, carbohydrates include sugars and sugar polymers. So as you know, every polymer is made of a bunch of monomers. So in this case, in the carbohydrate world, a monomer is called a monosaccharide. Let's talk about that. So monosaccharide, there are 2 C's in there. Saccharide is an interesting spelling word. But whenever you hear the word saccharide, think sugar. That's something sugary sweet. Mono is obviously 1, so you have 1 sugar. And so a definition of a monosaccharide, this would be the monomer for carbohydrates. So this would be a carbohydrate monomer. The most famous of the monosaccharides is glucose. So glucose is something our body utilizes for energy all day long. That's how our cells can perform all the functional work they need to do. We utilize glucose very, very well. So no doubt that our studies in carbohydrates is going to focus on glucose a lot. So let's talk about glucose. Glucose is C6H12O6, a 6-carbon chain. And glucose is the most common monosaccharide. Most common monosaccharide. Now, glucose can exist either as a carbon chain or as a carbon ring. I'm going to show you both structures, but what you need to know is that glucose prefers being in a ring shape when it's creating all of its polymers, and we'll talk about that as well. So let's do 1, 2, 3, 4, 5, 6. Make that a little bit nicer looking for you. Hang on. This is what glucose looks like in its 6-carbon chain. And as I said, glucose likes to be within a ring when it makes all these polymers. So this is the structure I want you to get used to seeing, because this is the functional form of glucose in our carbohydrate world. So that oxygen this oxygen is this double bond oxygen right here. What happens is these carbons just chain up right at that double bond O. So you have a carbon, you have 1, 2, 3, 4, 5, 6 carbons in this ring. And of course you have all your functional groups on there as well. So for glucose, you've got your OH groups. Go. That is glucose right there for you. And actually, that's fine. You want to be a little bit more precise about it. These can actually flip around, but oftentimes you'll see it like this as well. Okay, there's your glucose right there. So that's what glucose looks like when it's in a chain or when it's in a ring. And in typical nomenclature for carbon chains, even when they're in a ring, we usually number the carbons. So what you need to know is that this oxygen is kind of where we start our numbering, but you don't number oxygens, you only number the carbons. So this first carbon over here, this would be carbon number 1, this would be carbon number 2, carbon number 3, carbon number 4, this one right here on the corner is 5, and this one is 6. So we still maintain our 6 carbons, and these get numbered as well. So that's what I want you to know about glucose. So that's when it's in its monomer form. So if you have a monosaccharide, if you know your prefixes well enough, you could guess how we're going to be building these polysaccharides. So if you put 2 monosaccharides together, you can get what's called a disaccharide. And the disaccharide world is something very common to us. That's where we get table sugar, maltose. We're going to talk about all these different things. So now that we know our world of monosaccharides, and glucose is the one that we care about the most, we're going to focus on glucose. But let's briefly talk about disaccharides. So big letter B, I'm going to erase all this. Big letter B was going to be disaccharides. So if we're putting together 2 monomers, what kind of reaction do we need to put 2 monomers together? You have 2 options. It's either a dehydration reaction or a hydrolysis reaction. It's a dehydration reaction. Very good. It's when you put 2 monomers together, you are dehydrating those 2 compounds of 1 water, right? So in order to make a disaccharide, this is the combination of 2 monomers of 2, I'll say 2 monosaccharides via a dehydration reaction. All right, now here's where it gets really fun, because when 2 monomers come together, they create a disaccharide on the other side, but the link between the 2 is very unique. So let's show what that looks like really quickly. So we're going to put 2 glucose monomers together. So let's draw 2 glucose rings. And draw another one. So we can put them together. Look good. All right. So here we have glucose plus glucose. And I want these 2 to react by a dehydration reaction. I want to bring these 2 molecules of glucose together. So remember, what gets produced during a dehydration reaction? What lovely hydrated molecule gets produced? We get water. So if you look at these 2, you basically want to squish them together. And remember, we use our functional groups to do the reactions. The functional groups are the ones involved in all this. And in this case, we have 2 hydroxyl groups basically facing each other on the glucoses. So what happens is this OH group wants to grab another hydrogen to create the water. It's not going to grab that hydrogen. It just doesn't. It likes to grab the hydrogen from a functional group because those functional groups like to react in the reactions. So this OH is going to grab the hydrogen over here. It could have been the opposite. This OH could have grabbed that hydrogen. It really doesn't matter as long as the 2 of them come together. Now when these 2 get together, you're going to get your water, right? So here's what it looks like on the other side. This is where you need to start picturing, if I were to squish these 2 together, what would this disaccharide look like? Here's my monosaccharide, put them together, I have a disaccharide. This oxygen that didn't react, that's key to linking these 2 together. So here's what it's going to look like on the other side. I'm going to shrink my arrow up just a bit. So here's my first glucose. And here's where it gets a little different. So here, this is the carbon where the reaction is happening. So this is my carbon right here. It's actually going to link, I got my hydrogen up here, it's going to link with the other oxygen. So this oxygen right here is this oxygen. So let's complete the picture, shall we? Okay, less water. That right there, you have glucose plus glucose, and when you do glucose and glucose together, you actually get a new monosaccharide, and this is called maltose. Right here. So ever heard of malt sugar? I love a good chocolate malt. If you sprinkle a little bit of malt sugar, that's maltose. That's what gives it not a super sweet taste. It's more like a diluted sweet feel to it, but I personally like it. So what you need to know is that this oxygen right here, after this hydroxyl group grabs that hydrogen from that second glucose, it pulls together this oxygen to link these two glucoses together. This linkage right here has a very special name, and I'm going to put a star next to it right here. That linkage right there is called a 1,4 glycosidic linkage. Let's write that down, and then I'll tell you why it's called that. So this is called a 1,4 glycosidic linkage. Take the water away for a second. So that 1,4 glycosidic linkage, here's why it's called a 1,4 glycosidic linkage. You got to get used to that glycosidic linkage part, but the numbering is important. That's telling you what carbons are involved in the linking together. So remember how we numbered the carbons in glucose? We always start at the oxygen and then work your way clockwise. So this oxygen right here, this will be carbon number 1, number 2, just got my hydrogen, number 3, 4, 5, 6. So this is carbon number 1 right here. So that's where the 1 comes from on this one. And you can probably figure out where the 4 comes from now. So if you number them, you got carbon 1, 2, 3, 4, 5, and 6. And there's carbon number 4. So you get your 1,4 glycosidic linkage. Glycosidic, just talking about the fact that we're linking together two glucose molecules. Once we get into the carbohydrate polymer world, you're going to see how the 1,4 glycosidic linkage is everywhere, because you're going to make chains and chains of this, really long chains. So this is how you do a dehydration reaction between two monosaccharides, in this case two glucose molecules, to create a disaccharide. Now you can imagine if we kept adding glucose and glucose and glucose and glucose, you can easily keep this dehydration reaction going forever if you wanted to. So let's really quickly discuss 3 important disaccharides. We already talked about one, which is maltose. So I'm going to erase all this, and we'll just list out 3 other disaccharides for you just to be familiar with. These are the common ones that you see a lot. So number 1, under this, would be common disaccharides. So the first one we already talked about. It's when you do glucose plus glucose gives you maltose. You know that one. You've seen it. The next one. You know very, very well from your sugar, essentially. When you put glucose with fructose, which is like fruit sugar, you get Glucose plus fructose gives you sucrose. That's table sugar. I was about to put sugar. No, it is sugar, but it is sucrose. You could also put table sugar if you want to know for later reference. Table sugar. This is the stuff that your parents always say, don't eat too much sugar. They're basically saying, don't eat too much of this disaccharide. You know, your body can only handle so much. And it's kind of true. But the difference between this sucrose, the white granular sugar that we're used to eating all the time, and high fructose corn syrup, it's a little bit different. High fructose corn syrup is not a disaccharide. It's literally glucose and fructose monomers in a viscous fluid, basically. That's the syrupy part of it. And the problem with high fructose corn syrup, they're not in 1-to-1 ratios. Whereas in sucrose, 1 glucose and 1 fructose put together gives you a sucrose. High fructose corn syrup, the ratio is a little bit off. You get a little bit more fructose. It's like 51% fructose to 49% glucose. Why is that so bad for you, you ask? I hate to say it, it's almost like what the commercials say. It's not bad in moderation, but it's really not the best thing for you. Our bodies are meant to take apart disaccharides really, really well. When you don't have to use the energy to take away a disaccharide and turn them into their monomers like this, you're basically allowing this to go straight into your bloodstream, and your body's not processing it as much as it possibly can. So it's interesting. The last one, is one of my favorite monosaccharide names. This is when you put glucose together with a monosaccharide called galactose. I just thought that's a cool name. So you have glucose plus galactose. And I don't know if you guys are seeing a pattern here, but every time you see something ending in OSE, all these oses, you know you have a sugar on hand. So in this case, glucose plus galactose, that's going to give you lactose, milk sugar. Lactose. Milk sugar. You or somebody you know may be intolerant to lactose. That means that they do not make the protein in their body, the enzyme that helps digest lactose into glucose and galactose. Our bodies are meant to break these apart, and sometimes there's some coding that goes wrong in our DNA that doesn't allow that to get broken down correctly. And so if you don't break down lactose, it hurts. So that's what you need to know about. These are your common disaccharides. So if anyone ever says, hey, what do you get when you mix glucose and fructose? You'll know right away, oh, that's table sugar, that's sucrose. Of course it is. So come back, we're going to learn on our next lesson, we're going to talk about the polymers of carbohydrates. And this is where it gets really interesting because we're going to start talking about different structures within plants and animal cells. So come on back.